Okay, so we are studying on genetics. As I said before, it is actually one of my favorite topics in biology because, after all, a lot of the more current, up to date research in biology are related to genetics. And we will learn more and more different kind of genetic techniques. After all, you hear things about on the news like, oh, so such and such researchers have discovered this thing which is related to the genes. You don't hear much news about scientists have reclassified a certain organism into a different group or family. You don't really hear it that much. Okay, so it's really exciting. We're gonna start talking about Gregor Mendel. Now, if you watch the video, and I'm teaching it presuming you have watched the video, Gregor Mendel did his experiment back in 1800s. We have done a lot of things since then. So in this case, some of the stuff that we're going to be talking about will be relatively simplistic. Okay, will be relatively simplistic. So in this case, um, do you think so keep that in mind as we're going on with this lesson. Next week we're going to start talking about more and more slightly more complex scenarios. Okay, now, and also one thing about today, the main point about uh, Mendel's experiment, this lesson is we kind of briefly learned about what did he do, because in some ways he was experimenting on plants and we just came from a plants unit. So in some ways it's a good segue. Another thing is you learn a lot of terminology and conventions. In this new unit, again, new definitions, new terms. Also, in genetics, you will actually solve word problems. Okay, so in this case, you kind of do need to understand, like when someone say crossbreeding, what does that mean? Okay, or test crossing, what does that mean? Um, when we're talking about P1 generation or F1 generation, what does that mean? So you have to know what these terms. Okay. Any questions? All right, so Gregor Mendel, Monk, 1900s, experiment on peas. Now, couple of things. So in this here, you notice why some of the pea plants that they have different traits. Okay. Now here the traits, the word traits, make sure you underline it because that's a terminology that you do make sure you do need to know. Okay. So when we're talking about traits, we're talking about a version of a characteristic that is inherited. Okay. This unit, there's going to be talking. There's always a lot of this idea. It's like, oh, is it nature or is it nurture? Is it something that's inborn due to information passed on from parents?
So like others, right?
Pardon?
So like something we know, like even when we're born, like reading.
Reading, you say?
No, something like something like that we naturally know, right?
Well, we're talking more like through the DNA.
Yeah.
Yeah.
So like it'd be a trait like similar to breathing that we already know how to do.
Yes, that we need to be able to breathe. For example, we're not like we can't. We're not exactly like facilitated aerobes. That will be interesting. We're obligate aerobes, so to speak. Okay. And in terms of other things like environmental or through learned behavior. Okay, so we're talking about things that's passed down from DNA. So traits is the particular version of characteristics. So let's just say go back to this example here. He was working on peas. He noticed some of them are tall, some of them are short, some of them have yellow seeds, some of them have green seeds. Okay, so those will be different versions of those traits. So in some ways, you have a gene that codes for the height. Okay, so there's a gene that codes for the height. There's a gene that codes for the color of the seed. But whether you get the green version of that gene or the yellow version of that gene, that's a different version. Are we good so far? Okay. So traits, that's important to know, particular characteristic. Now, Brigham Mendel, again, heredity, what does that mean? Okay. The passing down of traits through the generation, and genetics is a study of that. Okay. Genetic is a study of hereditary. So make sure you know these definitions. Okay. Any questions so far? Okay. I'm just going to go back to here simply because it's nicer, bigger, and colored. The scientific process that he went through. Now, I'm not going to spend too much time on the scientific process that he did. I'm going to use it as an example to talk about certain terms and what did he discover. Okay, so some of the stuff that you do need to know. You do need to know that he experimented with pea plants. Okay, there will be a test question of sort asking what on earth did he experiment with pea plants. Okay, and in some ways he was very lucky, so to speak, and I'll explain why he was so lucky. Because the traits that he was looking at doesn't have any, it only has a alternate form. So it's either yes or no, green or yellow, tall or short. You don't get like a blend. Okay, you don't get a blend of like tall plant cross with short plants give you medium plants. Okay, you don't get those. Okay, so here in this slide, you see a number of things. So seed shape, seed colors. Okay. So the shape of the pea, whether it's smooth or wrinkled, yellow or green, flower color is only two form, position of the flower, pod color, pod shape, and plant height. Okay. He was very lucky. A lot of scientists later on, when he looked back at analyze his experiment, one thing they comment is like, he was very lucky. He happened to pick a species where these traits is either this or that. There are some other traits, even in the human system, like that is much more complex. Okay. For example, we have type A blood, we have type B blood. We also have a type O blood and type B blood. Luckily, he didn't chance upon like organisms that have or traits that is that complex. So he was very, very lucky. Okay. Again, in a week or so, we'll start looking at these like slightly more complex scenarios. Right now, we're just learning the basics, conventions, and rules. Are we good? Okay. So couple of things. He did experiments, has controlled. I'll skip that part. Now, here, this term you do need to know. Okay, when you talk about developing a true breeding or pure breeding stock, you are basically talking about plants that will have the same trait from parents to offspring for many, many generations. Okay, true breeding stock or pure breeding stock means they are purebred. They will always, generation wise, they will always have certain conditions that are the same. Now, how on earth did he accomplish that? Well, remember flowers have like the filaments and the pollen sac on in the flower, and you have the stigma, which leads to the female gametes. So what he did was either he self-pollinated the plants, okay, using its own pollen and own stigma to pollinate it, so it will have basically not quite clone itself, but reproduce with itself, or they will cross it with another plant that is also purebred, okay, true breeding plants. So this is basically how he did it. Okay, are we good? Yeah. So here the slide talks about how did he self-pollinate and cross-pollinate the plants, okay? You do need to know the two definitions. What does that mean? Self-pollinate and cross-pollinate. Okay, make sure you do know those terms. Okay. Now, going back to his experiment. So one of the experiments that he did was he was pollinating the plants from a purple plant crosses with a white flower. Now, so this convention here, the crossing, basically in some ways think of mating. Okay, think of mating, but this is generic. You can talk about plants, you can talk about animals. Okay, so because he was cross-pollinating, we also use the X cross as like these two are the parents. Okay, so in this case, the parents generation, we use a P generation for short. Okay, and what later on happened is this. So he was crossing a purple flower plant with a white flower plant. What he found out later on is this. He noticed that all the offsprings have purple flower. Okay, so all the offsprings will have purple flower. So this here, this is the purple plant, this is the white plant, this is the parents generation, the P generation. These first generation offspring will have F1 generation. Okay, F1 generation. Yes.
When you say cross-pollinate, does it have to be between like
two individuals?
Two individuals, but can they be of the same species?
But they have to be the same species. Yeah, we're not talking about like cross species. Yeah. Yes.
Can you explain true breeding and pure breeding?
True breeding and pure breeding, they're both the same, different way to say the same thing. Basically by in that case, he was self-pollinating the plants for over generations. So essentially all the offsprings from generation to generation will have the same trait for same certain traits. Okay, could be always white flowers, could be always tall plants. I don't know what. Okay, so you just need to know the terms for that. We do that too in a lot of like lab animals wise, so to speak. But we'll talk about that later. Okay. Questions? Yes.
When you talk about true breeding or pure breeding stock, is it always like self-pollination or like crossbreeding with a similar plant?
Both, either or. Yeah. The method of how to attain that is either or. Either self-pollinate. In plants, you can easily do that. Animals, not easily. But in animals, would be like you bred it with another individual that has very similar genetic traits. So, for example, you can go to a scientific catalog and order a certain kind of mice that they will actually have they will say like they will have this version of this gene. How they get that would be just basically always getting this mice to cross with its sibling and then generation after generation. I know. Yeah.
Thank you.
Yes. Yes. Oh, we good? Okay, so we're talking about first generation. So the parents' generation is P, and you see the two individual crosses, and then all these offspring are first generation. So in this case, what they notice is this: all the first generations are purple plants. Okay, we're gonna come back to that. He also noticed a couple of scenario that is interesting, simply because. So, for example, you notice that you have tall plant crosses with short plants. All the first generation offspring will be tall. Green pots versus yellow pots, all the first generation will be green. And same thing for the yellow pea plants versus crossing with green pea plants. Okay, so he noticed some of these things. Why is this significant? It says about that in the video. Yes, I think it said about that in the video.
Does it have something to do with the dominant and?
It does relate to dominant and recessive, yes, but something else. What do people think about hereditary at the time of Gregor Mendel? Yes.
They thought that
Yes.
Disease?
What?
Diseases? Like, I mean, like when if a parent had like cancer or something, they were concerned about whether it would be like something hereditary passed down to future generations.
I don't know if people thought about it back then. It could, but that wasn't what I was looking for.
is it related to like they wondered how if their children had a trait, for example, if it was like both parents had a certain trait and then suddenly out of nowhere they got some trait that none of their parents had. It was like they wondered where that trait came from. And if they thought it was a legitimate child.
No, I wasn't thinking about that. That's not also another thing that I wasn't thinking about. I was thinking about that back then. A lot of times they were thinking about usually the two parents, the trait will actually just mix, and then the offspring will have like a medium, so to speak, like the tall and the short. Tall parent, short parent, all the offspring will be medium. But having this shows that, wait, genetics is not as they were thinking about at the time. So with this later on, we will talk about what Matthew, what you're saying about the dominant and recessive. In fact, this is where we're going with that. Now, a couple other things. He also took these first generation offspring and either self-pollinate or cross-pollinate with other first generation offsprings. Okay, and so in this case, the generation at that point, the second generation, you call it F2 generation. Okay, you call it the F2 generations. Okay, so parents. F1 it's the first generation of offspring. F2 will be the second generation offspring. And what he noticed is this: there's always some interesting ratio. Okay, there's always some interesting ratio. So, for example, purple plant crossed with a white flower plant, all the first generation offspring will be purple. But then if you take two of these and cross with each other, it would be about 3-to-1 ratio of purple to white. Okay. Same thing for tall and short plants. Okay. By second generation, there will be about 3 tall plant to 1 short plant ratio. Okay. And if you look at the numbers here, he wasn't even doing like a small number of offspring to guess. He basically, if you look at this slide here, he did in hundreds or thousands, okay? 700 to 224, okay? In that kind of calculation. So he was doing with a lot, really large sample size to establish these ratio, okay? Are we good? So he kind of did a lot of experiments. He established certain ratio to kind of come up with certain rules. Now, a couple of terms that we really should know. When he talked about factors, we're talking about traits. This is what we currently call genes, okay? Back then, they didn't know DNA, so they didn't really use the word genes at the time. So what he would say is different factors determine a trait. So if one factor of the gene is dominant, it masks the effect of the recessive trait. Okay? So using flower color for an example again, the fact that when you have a purple flower crosses with a white flower and all its offspring, first generation offspring, are purple flower, it would show that purple flower would be the dominant trait, okay, the dominant gene, and the white color trait would be the recessive trait, recessive gene. Are we good? So you have one gene for the flower color for peas. You have the purple version or the white version. The purple version is dominant, the white version is recessive. Good? So Mendel's law of segregation. Mendel law of segregation. This is another name, another rule that you really should know. So what he establishes is each trait, each characteristic, flower color, carries two factors or two genes, one from each parent. Okay, one from each parent. So all those first generation pea plants, they will have one version of the purple, one copy of the purple version gene from one parent, and the white version color gene for the other parents. Okay, so each pea plant will have two copies of that same gene, same traits. The parent donate only one copy of each gene in their gametes, sperm or eggs, pollen or ovule. Okay? During meiosis, two copies of each gene separate or segregate. Okay. We briefly talked about meiosis back then when we were looking at fungi and protists and all those stuff. Okay. We will kind of go through that a little bit more in the next few lessons. Okay. So that's Mendel's law of segregation. Now, a couple of things. We're going to start off talking about monohybrid cross. When we're talking about monohybrid cross, we're just talking about one trait, okay? Crossing parents of two parents on one particular trait. And they're going to be relatively simple, like where it's an on or off kind of scenario. Okay? Now, we're going to use these things called Punnett squares. The purpose of Punnett square is to predict the probability of outcomes, the probability of in this case, what's the chance of the offspring having this particular trait? Okay. A real life, any questions so far? A real life example. My wife and I are good friends with this couple, and this couple, they're hoping to have kids. because of their of certain demographics, they decide to like, hey, let's go to a genetic counselor to check something out. And what they found was that they're both carriers of the sickle cell anemia gene. Okay, sickle cell anemia gene, so they don't have sickle cell anemia, but they carry that gene. And if they have kids, 1 out of 4 chances their kids will have sickle cell anemia. And if they don't have sickle cell anemia, 2 out of 4 chances their kids will also be carrier of that gene too. And they really only have one out of 4 chances that the kid will not have sickle cell anemia and will not be carrier. Okay, so the genetic counselor figured that out by doing Punnett squares. Okay, so this is like how Punnett square works in predicting the probability of certain traits coming up. Okay, are we good? Okay, we'll learn about sickle cell anemia probably in the next week or so. It is a genetic condition that affect largely on certain demographics more than others. Okay, are we good? Okay. Another thing, alleles. Okay, this term alleles. Alleles is you're talking about the different form of certain traits. So for example here, a human example would be like curly hair or straight hair. Okay, curly hair and straight hair. Okay, they're both different form, okay, of the hair texture gene. So all of us will have a gene that codes for our hair texture. Okay, whether we have the version that codes for straight hair or the version that codes for the curly hair. Okay, so there are 2 different versions of the gene. Okay, so when we're talking about allele, they're talking about different version of the same gene. Now, if both version that you got are the same, like let's just say you got both gene from your parents are both straight hair gene or curly hair gene. If that's the case, you have what you are considered homozygous. Okay, homozygous individual for that trait. If you have one copy of curly and the other copy of straight from the parents, you will be called heterozygous for that gene. Okay? Homozygous, 2 copies of the same. Heterozygous, different copies. Okay, are we good? Now, oops. Okay. Yes.
So then these alleles, like, does it relate to only the heterozygous, or does it have to do with both?
It does with both. So basically, alleles are the different form of the gene, right? So whether you're talking about pea flower colors. So there is a gene that codes for the color color of the flower. So you have the version of purple or white. Okay, 2 forms of the same gene for the color flower color. Hair, you have a gene that codes for hair texture, curly or straight. Okay, so that's allele. So if both alleles that you got from your parents are either both straight hair or curly hair, that's your homozygous for that trait. If one is different from the other, heterozygous. Good? Okay. Are we good here? All right. Now, so in this example here, you see the purple flowers you're either homozygous. These are capital P's. You could have a capital P and the lowercase p. Those would be heterozygous. And white color would be homozygous lowercase p. Okay. Also, this leads to another set of terms. Genotype versus phenotype. Genotype is what kind of an allele an individual has. So we're talking about either big P, small p, or two big P or two small p's. When we're talking about what kind of letters or what kind of alleles, that's genotype. Phenotype would be the outcome or the outward appearance of that trait. So instead of talking about whether it's uppercase P or lowercase p or big P, small p's, you're talking about purple color or white color, curly hair or straight hair at the end. Okay. Are we good? Yes. So genotype, when we're talking about genotype of a certain individual, you're talking about like what kind of allele it has. Because, for example, here, you could have two copies of capital P or one copy of capital P, one copy of lowercase p, okay? Homozygous, heterozygous, but these situations, they will turn out to be purple flower anyways. So what type of allele you have, that's called genotype. What kind of outward appearance of that trait, that's phenotype. Okay? So typically when we're talking about homozygous or heterozygous, you're talking about genotypes. Okay? Are we good? Now, last but not least, this thing called test crossing. Test crossing. Now, test crossing is used to determine if a individual whose phenotype is dominant, whether you want to figure out is it homozygous or heterozygous. Okay? Homozygous or heterozygous. So coming back to the pea plants here, the pea colors, flowers right here. So let's just say you have a purple flower plant. You don't know whether it's a two big peas or a big pea, small pea. Okay? You don't know. What you do is do test crossing. By test crossing is you're going to cross it. Okay? So this unknown, you'll cross it with a homozygous recessive genotype. Okay? So you'll cross it with a flower that is homozygous lowercase p or a white flower plant. Okay? Now, if the purple flower plant that you don't know about was a homozygous plant, so which means both uppercase p, then all the offspring will be dominant, will also be purple flower. Okay. So if the offspring from the cross result in all dominant phenotype, then the unknown is dominant. Okay. Actually, sorry, this should not be dominant. This should be homozygous. Homozygous. Okay? Now, if you look at the first generation offspring, from that test cross and you see both purple flower and white flower, dominant and recessive traits, then The flower that you're not sure about would be heterozygous. Okay? If offspring include both dominant and recessive, then the unknown is heterozygous. Okay? So in this next slide here, you have a purple flower plant. You cross it, you test. You don't know whether it's homozygous or heterozygous. You have one big P, you just leave it as a blank. So you don't know. So you cross it with a white flower plant. The white flower plant is homozygous. Both are small peas. So you end up two scenarios. If this is a homozygous purple plant, so two big peas, all the offspring should be purple color. If this is one big pea and one small pea, so it's heterozygous purple, half the plants, half the offspring should be purple, the other half should be white. Okay? Does that make sense? This will make more sense when we start doing Punnett square problems. It will be quite clear. Okay. Are we good? Okay. That's about it. So, a couple of things. Now, tomorrow I'm not going to be here. There is a new teacher meeting, so I'm going to a new teacher meeting. Okay, so I won't be here. So there will be a supply teacher. So a couple of things. First, you will need to bring your textbook. I kind of wrote down what you will be needing to do already. There will be 2 sets of handout from the teacher. I'll leave them with the teacher. I think it's the same one from last Friday. Okay. So there will be 2 sets of handouts. You're to read them and answer some of the questions from the textbook. You will also, last day of collecting your data and observation. Okay? Yes?
Let's please water the bags with soup and water.
I meant wash the bag with soap and water. Okay? This is what happened when you do autocorrect but you didn't proofread. Okay? One of those scenarios.
Mr. Chang,
Yes.
do this question?
Not right now. Okay? I think it will make more sense when we start doing more stuff. Okay? Any questions? So make sure you bring a device. Okay? Bring your textbook. Okay? Unfortunately, I can't book the Chromebook because OSSLT. All right? Any questions? Okay, so